let's delve into the world of mitochondria, which are probably my favorite organelle. So let's just have a little review of what mitochondria are, and then we can delve a little bit deeper into their structure. So let's just think about a cell, and not just any cell, but a eukaryotic cell. So that's the cellular membrane. And when people say a eukaryote or a eukaryotic cell, they most typically say, oh, that must have its nuclear DNA in a membrane-bound nucleus. And that would be true. So let's draw our membrane-bound nucleus. That's our nuclear membrane. You have your DNA in here, so let's draw some DNA. But when we talk about eukaryotic cells, we're not just talking about a membrane-bound nucleus. We're also talking about other membrane-bound organelles. And in a close second place for a membrane-bound structure that is very important to the cell would be the mitochondria. So let's draw some mitochondria right over here. So I'll talk a little bit more about what these little squiggly lines that I'm drawing inside of the mitochondria are. This is actually a little bit more of a textbook visualization, as we'll learn in a few minutes or seconds that we now have more sophisticated visualizations of what's actually going on inside of a mitochondria. But we actually haven't answered all of our questions. But you might have already learned that, so let me make it clear. These are mitochondria. That's the plural. If we're just talking about one of them, we're talking about a mitochondrion. That's the singular of mitochondria. But you might have already learned sometime in your past or in another Khan Academy video that these are viewed as the ATP factories for cells. So let me write it this way. So ATP factories. ATP factories. And if you watch the videos on ATP or cellular respiration or other videos, I repeatedly talk about how ATP is really the currency for energy in the cell, that when it's in its ATP form, you have adenosine triphosphate. If you pop one of the phosphate groups off, you pop one of the P's off, it releases energy. And that's what your body uses to do all sorts of things, from movement to thinking to all sorts of things that actually go on in your body. So you can imagine mitochondria are really important for energy, for when the cell has to do things. And that's why you'll find more mitochondria in things like muscle cells, things that have to use a lot of energy. Now, before I get into the structure of mitochondria, I want to talk a little bit about its fascinating past, because we think of cells as the most basic unit of life, and that is true. That comes straight out of cell theory. But it turns out the most prevalent theory of how mitochondria got into our cells is that at one time the predecessors, the ancestors to our mitochondria were free, independent organisms, microorganisms. So they're descendant from bacteria-like microorganisms that might have been living on their own, and they were maybe really good at processing energy, or maybe they were even good at other things. But at some point in the evolutionary past, they got ingested by what the ancestors of our cells, and instead of just being engulfed and being torn to shreds and kind of being digested and eaten, it was like, hey, wait, if these things stick around, those cells are more likely to survive because they're able to help process glucose or help generate more energy out of things. And so the cells that were able to kind of live in symbiosis, have them kind of give a place for the mitochondria to live, or the pre-mitochondria, the ancestor mitochondria, those survived. And then through kind of the processes of natural selection, this is what we now associate. We now associate eukaryotic cells as having mitochondria in it. So I find this whole idea of one organism being inside of another organism in symbiosis, even at the cellular level, that's kind of mind-boggling. But anyway, I'll stop talking about that, and now let's just talk about the present. Let's talk about what the actual structure of mitochondria are. And I'll first draw kind of a simplified drawing of a mitochondrion, and I'm going to draw a cross-section. So I'm going to draw a cross-section. So if we were to kind of cut it in half. So what I've drawn right over here, this would be its outer membrane. This is the outer membrane right over here. Let me label that. Outer membrane. And all of these membranes that I'm going to draw, they're all going to be phospholipid bilayers. So if I were to zoom in right over here, if I were to zoom in, we would see a bilayer of phospholipids. So you have your hydrophilic heads facing outwards, and your hydrophobic tails facing inwards. So So you see something just like that. So they're all phospholipid. They're all phospholipid bilayers. But they aren't just phospholipids. All of these membranes, they have all sorts of proteins embedded. I mean, these cells are incredibly complex structures, but even organelles like mitochondria have a fascinating, I guess you'd say, substructure to them. They themselves have all sorts of interesting proteins, enzymes embedded in their membranes that are able to help regulate what's going on inside and outside of these organelles. And one of the membranes that you have in, or one of the proteins that you have in the outer membrane of mitochondria, they're called porins. And porins aren't found only in mitochondria, but they're kind of tunnel proteins. They're structured so they kind of form a hole. They form a hole in the outer membrane. So I'm drawing them the best that I can. These are porins. And what's interesting about porins is they don't allow large molecules to pass through passively, but small molecules like sugars or ions can pass passively through the porins. And so because of that, your ion concentration and your small molecule concentrations tend to be similar on either side of this membrane, on either side of this outer membrane. But that's not the only membrane involved in a mitochondrion. We also have an inner membrane. I'll do that in yellow. We also have an inner membrane, and I'm going to draw it with the textbook model first, and then we'll talk about a little bit about what, since we now think this model is not quite right. But in this, so we have this inner membrane, inner membrane, and this inner membrane has these folds in it to increase their surface area. And the surface area is really important for the inner membrane because that's where the processes of the electron transport chain occur across essentially these membranes. So you want this extra surface area so you can essentially have more of that going on. And these folds have a name. So if you're talking about one of them, if you're talking about one of these folds, you're talking about a crista. You are talking about a crista, but if you're talking about more than one of them, you would call that a cristae. Cristae, sometimes I've seen the pronunciation of this is cristae or cristae. That's plural for crista. And these are just folds in the inner membrane. And once again, the inner membrane is also a phospholipid bilayer. Now, inside of the inner membrane, so between the outer membrane and the inner membrane, you could imagine what this is going to be called. That space is called the intermembrane space. Not too creative of a name. Intermembrane space. And because of the porins, the small molecule concentration in the intermembrane space, and then outside of the mitochondria, out in the cytosol, those concentrations are going to be similar. But then the inner membrane does not have the porins in it, and so you can actually have a different concentration on either side, and that is essential for the electron transport chain. The electron transport chain really culminates with a hydrogen ion gradient being built between the two sides. And then they flow down that gradient through a protein called ATP synthase, which helps us synthesize ATP. But we'll talk more about that maybe in this video or in a future video. But let's finish talking about the different parts of a mitochondrion. So inside the inner membrane, you have this area right over here is called the matrix. It's called, let me use this in a different color. This is the matrix, and it's called the matrix because it actually has a much higher protein concentration. It's actually more viscous than the cytosol that would be outside of the mitochondria. So this right over here is the matrix. And when we talk about cellular respiration has many phases in it. We talk about glycolysis. Glycolysis is actually occurring in the cytosol. So glycolysis can occur in the cytosol. Glycolysis. But the other major phases of cellular respiration, when we talk about the citric acid cycle, also known as the Krebs cycle, that is occurring in the matrix. So Krebs cycle is occurring in the matrix. And then I said the electron transport chain, which is really what's responsible for producing the bulk of the ATP, that is happening through proteins that are straddling the inner membrane, or straddling the cristae right over here. Now, we're not just done, and probably one of the most fascinating parts about mitochondria. We said that we think that they are descendant from these ancient independent life forms. And in order to be an ancient independent life form, they would have to have some information, some way to actually transmit their genetic information. And it turns out mitochondria actually have their own genetic information. They have mitochondrial DNA. And they often don't just even have one copy of it, they have multiple copies of it. And they're in loops, very similar to bacterial DNA. In fact, they have a lot in common with bacterial DNA, and that's why we think that the ancestor to mitochondria that lived independently was probably a form of bacteria or related to bacteria in some way. So this right over there, that is the loop of mitochondrial DNA. So all of the DNA that's inside of you, the bulk of it, yes, it is in your nuclear DNA, but you still have a little bit of DNA in your mitochondria. And what's interesting is your mitochondrial DNA, your mitochondria are inherited essentially from your mother's side, because when an egg is fertilized, a human egg has tons of mitochondria in it. It has tons of mitochondria, and I'm obviously not drawing all of the things in the human egg. It obviously has a nucleus and all of that. The sperm has some mitochondria in it. You can imagine it needs some energy to be able to win that very competitive fight to get to fertilize the egg. But the current theories are, well, most of that gets digested and dissolved once it actually gets into the egg. And anyway, the egg itself has way more mitochondria. So the DNA in your mitochondria is from your mother, or is essentially from your mother's side. And that's actually used, mitochondrial DNA, when people talk about kind of an ancient Eve or tracing back to having kind of one common mother, people are looking at the mitochondrial DNA. So it is actually quite fascinating. Now, I said a little bit earlier, and obviously it has its own DNA, and then because it has its own DNA, it's able to synthesize some of its own RNA, its own ribosomes, so it also has ribosomes here. But it doesn't synthesize all of the proteins that are sitting in mitochondria. A lot of those are still synthesized by, or coded for by your nuclear DNA, and are actually synthesized outside of the mitochondria and then make their way into the mitochondria. But mitochondria are these fascinating things that they're all, they're these little creatures living in symbiosis in our cells, and they're able to replicate themselves. And I don't know, I find all of this mind-boggling. But anyway, I said that this was the textbook model, because it turns out when you look at a micrograph, a picture of mitochondria, it seems to back up this textbook model of these folds, these cristae just kind of folding in. But when we've been able to have more sophisticated visualizations, it actually turns out that it's not just these simple folds, that the inner membrane essentially pokes into the matrix, and it turns out it has these little tunnels that connect the space inside of the cristae to the intermembrane space. So I like to think about this because it makes you realize, we look in textbooks and we take these things like mitochondria for granted. Like, oh yeah, of course, that's where the ATP factories are. But it's still an area for visualization and research to fully understand exactly how they work and even how they are structured. That this baffle model, where you see these cristae kind of just coming in and out of the different sides, this is actually no longer the accepted model for the actual visualization, the structure of mitochondria. Something more like this, something more where you have this cristae junction model, where you have, if I were to draw a cross section, where I drew the outer membrane, the inner membrane, I'll just draw, has these little tunnels to the actual to the space inside of the cristae. This is actually now the more accepted visualization. So I want you to appreciate that, when in biology you read something in a textbook, you kind of say, oh, people have figured all of this stuff out, but people are still thinking more about, well, how does this structure work? What is the actual structure? And then how does it actually let this organelle, this fascinating organelle, do all of the things that it needs to do?